Hello, guys. Welcome you all to Vedora, the aura of knowledge, radiance of wisdom. So, beginning with the modern topics, we begin with the physiology of homeostasis. So, this session is gonna be full on research, which I have done on the previous year question papers to just make it simple for the exam purpose. So, knowing the exam pattern is actually the half battle one. So, this chapter is gonna come either for 10-mark question or as a 5-mark question, but sure, you'll be getting one MCQ from this chapter. So, you can prepare according to it. So, for each topic, I have been done this PDF in a form of answer format only. You can just give it in that way itself, how I have given. So it will be helpful for you in notes taking also. So see that everyone have their own notes. That is more important for self study. So passing on to the concept, homeostasis. So what is homeostasis? Let us break down this homeostasis into two. Homeo and stasis. Homeo means same and stasis means which is standing or which is resilient. So it refers to a constant internal environment. Coming to the mechanism of homeostasis, so it is actually for the maintenance of the normalcy of the body. Actually, in our body, each and every second the body keeps changing. It is not the same at all the time. But then what does this normalcy mean? The one which does not cause any harm or disorders to our body is normalcy. The changes may be there, may be present, but they do not cause any abnormalities. Okay, so any change in a system behavior is regulated by the effectors and through the feedback signals which either reverse the change or it enhance to restore the normalcy. So these are called the feedback signals. So how is this homeostasis restored through the feedback signals? So feedback is what? It is a process, it is an output signal of the body to just bring our body to the normal condition. So coming to this, we have two types of feedback, negative and positive feedback. On seeing each on detail. So, what is negative feedback? So, here it is a system which arrests the change or reverses the direction of change. For example, we have body temperature regulation. So, when you have a high body temperature, what happens? The body temperature is sensed by the receptors present in the extremities. So this is reached to the brain or higher control centers. That's the CNS. And then from there it goes to the effectors. The CNS sends the command to the effector. So here the blood vessels and sweat glands are activated. Then the negative feedback happens. So what is happening? Increased blood flow to skin and increased sweating stimulus is removed, so therefore the homeostasis is restored. So like this. So what is high is being brought down. So that is negative feedback. For example, I'll give you another example. Uh, let's take thyroid hormone. So when you have a decreased secretion of thyroxine, what happens? Our body, to bring back the normal level of thyroxine, it does some action. That is, it stimulates the TSH secretion. Then, on stimulating the TSH secretion, it increases the secretion of thyroxine, by which the normal level of thyroxine is restored. If this condition is opposite, that is in increased case, what happens? It will inhibit the TSH secretion and it will decrease the thyroxine secretion too. So this is what is called negative feedback, that is decreasing something, the production of something to restore the homeostasis. Coming to positive feedback, so it is to increase the intensity in the same direction it goes. Okay. So for example, Here they have given the reflex of parturition. So when the head of the baby is pushing against the cervix, what happens? The cervix is getting some nerve impulses, and these impulses are transported to the CNS, that is the brain. From brain, the pituitary gland secretes the oxytocin, and this oxytocin is carried through the bloodstream to the respected area, and the effector, that is the uterus, it stimulates the uterine contraction and then it pushes the baby towards the cervix. So this is a contraction reflex in parturition. And another example can be clotting. So in blood clotting, what happens? Injury to blood vessels and bleeding may happen. Due to that, the formation of prothrombin and activators will happen. Then conversion of prothrombin into thrombin, then conversion of fibrinogen into fibrin, and then the blood is stopping. So here in this process, in positive feedback, something is being created here, okay, and in negative feedback, something is being offed, okay. So this is the major difference. So examples of positive feedbacks are like milk ejection reflex, parturition, and blood clotting. And in negative thing, you have regulation of secretion of hormones and water balance. Passing on to the next one. Next, we have a concept called feed forward control system. What is it? So it is actually an anticipatory. That's a predictive control which is not reactive. It does not depend on the feedback form of the end result. So the goal is actually to minimize the disturbances even before their effect in our body. Coming to some examples, salivation reflex, let it be, or preparation of a digestive tract for food, exercise anticipations, then thermoregulations on putting on warm clothes before going out in cold weather, like that, and digestive secretions. Then, actually, when our body is going for an exercise, we do some pre adaptations like warm up, that comes under free forward control system. And then secretion of gastric juices in digestion. An increase in secretion of luteinizing hormone prior to ovulation is also a type of feed forward control system. So these are all the examples. Passing on to the next topic, homeostatic imbalance. So the imbalance in this homeostasis is actually the homeostatic imbalance. So what is that? The constant internal environment is disturbed. Okay, it is out of our control. So that is called homeostatic imbalance. So when these systems malfunction or are overwhelmed, imbalance occurs which leads to disease and dysfunction. So, for example, in diabetes mellitus, the failure of glucose regulation is an example, and also and another example could be heat stroke. So, the failure of temperature regulation causes heat stroke. So, these are the example of homeostatic imbalance. Next, coming to an important topic, cell physiology and membrane physiology. So, this is actually a very vast topic which may be asked in 10 mark question. So mostly in this lesson, you may be getting 10 mark as homeostasis, explain homeostasis. In that you will be explaining all the three types, that is positive, negative and feed forward, along with homeostatic imbalance. Next topic will be the cell physiology and membrane physiology. In this we have the active transport, passive transport, their types, and it goes on like that. So now active transport. Coming to active transport, so, what is active transport? Already we have learned about it. So, something is being transported, what? The molecules across a cell membrane using some energy, that is ATP. There will be concentration gradient which is from lower to higher concentration. So, that is called active transportation. So here we have three types of carrier proteins for that. So what are these carrier proteins? They are a medium to carry these substances across the membrane. So we have three: uniport, symport and antiport. Uniport means moves one type of molecule in one direction. For example, when you have a calcium pump. Only the calcium is gonna move from one direction, that is from lower concentration to the higher concentration. Next is symport. So two different molecules in the same direction it goes. Sodium glucose symporter in intestinal cells. And antiport, we have the counter transport. Like it moves two different molecules in the opposite direction, like sodium potassium pump. Coming to the mechanism of it, here the substance binds to the carrier protein, then the ATP is hydrolyzed where the energy is released, then the carrier protein changes the shape, substances move across membrane against the gradient, substance is released on other side, carrier returns to original shape. So initially the ATP is needed, then the energy is generated from that, the carrier protein carries the thing, the shape changes, then the molecule transport happens. So the substrate transported can be ionic and non-ionic. Ions like sodium, potassium, calcium, hydrogen, chlorine, iodine can be transported. And non-ionic forms are like glucose, amino acids, and urea. Coming to the types of active transport, we have two, primary and secondary. In primary, what happens? The mechanism is like three sodium ion binds inside and then the ATP is hydrolyzed. Then the sodium ion is released outside. Then the potassium binds outside. Then the phosphate will be released. Then phosphate is released inside. So this is how the chain goes. So here 3 sodium ion is replaced by 2 potassium ion and 2 by 1 phosphate. So how this is how it goes on. Next is the secondary active transport. Here, the ion gradient is there from primary transport is carried to this. Then the carrier protein is involved in carrying the ions. Then the molecule is transported against the gradient. In secondary only you have symport and antiport. Symport is a core transport and antiport is a counter transport. So I have given it in the image so it can be useful for you. Next, coming to passive transport. So, passive transport is the movement of molecule across the cell membrane without the use of energy. That is the thing which used to be highlighted. So, which is from down their concentration or electrochemical gradient. So, here we have three types. Simple diffusion, that may be with a lipid layer, and facilitated diffusion through the protein layers or channels, facilitated or carried, then osmosis. Osmosis is through a selectively permeable membrane. So simple diffusion, here the molecules will move directly through the lipid layer. For example, like oxygen carbon dioxide transport during respiration, you have been studying it. So oxygen into cell, CO2 out of the cell, and also steroid hormones are being transferred through simple lipid layer. So some factors may affect this thing like concentration difference, molecule size, lipid solubility, and temperature. Coming to facilitated diffusion. So here this facilitated is also called as carrier mediated diffusion. So the high concentration binds the carrier protein, then transport through the membrane, then the low concentration is attained, then equilibrium will be formed. So here glucose transport can be happening, amino acid transport, iron transport and all are common. Then here you have protein channels, regulation of channels. So protein channels are calcium channels, sodium channels, potassium channels, and chlorine channels. And regulation channels are gated and ungated channels. Next is osmosis. So what is osmosis? It is a flow of water molecules across a semi-permeable membrane from an area of low solute concentration to a higher solute concentration. So this is how these are the types of passive transport. Coming to bulk transport, it is a type of transport in which large molecules or large amount of substance are transported through vesicles. So it requires energy, It is used for molecules of too large size for diffusion or channels. Now coming to types of bulk transport, we have endocytosis, exocytosis, phagocytosis, phagocytosis and receptor mediated endocytosis. So what is endocytosis? Means movement into the cell via vesicle, like phagocytosis of bacteria or phagocytosis of fluid it may be. Coming to exocytosis is movement out of the cell by vesicle. So endo means in and exo means out. Like secretion of neurotransmitters, hormones comes under exocytosis. Phagocytosis means cell eating, it is termed as. Uptake of solid particles like macrophages engulf the bacteria, which we have studied in 12th. Phagocytosis is cell drinking, uptake of fluid by the cell. Receptor mediated endocytosis, uptake of specific molecule by receptor binding, like LDL cholesterol uptake and all. Okay, what are the use of these terms? This may be present in your book. So mostly we all follow Shambulingam textbook. So I have made this notes based on that textbook. So even you can also follow it for better guidance. So these terms keep on occurring frequently in your texts. So to understand the concept better, you have to know all these terms. So for that purpose, I have included this. And rather than that, bulk transport is a concept which you have to know. When you go for further physiological studies, there may be terms regarding this. So for better understanding, you have to know this. Moving on to the next topic is resting membrane potential and the action potential. So what is resting membrane potential? It is the difference in the electrical potential across the cell membrane when the neuron or the muscle fiber is at rest. Okay, mark the point, it is at rest. Next, what is this action potential? It is a rapid reversal of the membrane polarity that propagates along the neurons or muscles. So, we have been studying the muscle contraction theory, sliding filament theory. So, in that, you will be learning about this resting and action potential. So, you will be having an idea about it. Coming to the basis, the electrical potential difference across the resting plasma membrane is called the resting potential. The electrical potential difference across the plasma membrane at the site of stimulus is called the action potential. So this is restricted to that site, and this is on the plasma membrane. That is the major difference. Coming to resting potential, neuron is not involved in conduction, whereas in action potential, neurons are involved. And neuron membrane is permeable to potassium ion and impermeable to sodium ion, whereas here it becomes permeable to sodium ion. Membrane is impermeable to the negatively charged protons of axoplasm and also chlorine ions, whereas here the membrane get deposited and becomes permeable to negatively charged protons of axoplasm. So if you have seen the graph of this resting membrane potential and action potential, resting membrane potential is around minus 70 millivolt. So it will be a straight line, whereas action potential is actually a curved graph. So that is the excitability and unidirectional conduction of both the things. Moving on to the acid-base balance. So this in Shambulingam book, it is not given under the homeostasis, that is cell physiology. It is given as a separate topic. But according to Ayurvedic NCSM syllabus, it is being included in physiology of homeostasis. So I have made it according to the syllabus, so it will be easy for you to go through. So what is acid base balance? It refers to a maintenance of the hydrogen ion concentration in the body fluid. So why it is essential? For the normal functioning of the body and metabolical activities. So here the normal pH of ECF, that is the extracellular fluid, is 7.4. It is maintained within a narrow range of 7.38 to 7.42. That zero point 0.2 variations can be accepted. Even slight deviation can impair physiological functions. The body has powerful mechanism to regulate the pH. So, what is hydrogen ion and pH? Hydrogen ion is actually a smallest ion particle and which is highly reactive. pH is defined as the negative logarithm of hydrogen concentration. So, actually, these things we have studied already in our school days. When pH is less, we call it as acidosis, and pH is more, it is alkalosis. That is, pH is less than 7 is acidic and more than 7 is alkalosis. So, a 1 unit charge of pH represents a 10-fold charge in H plus concentration. So, 1 is to 10 ratio is the pH versus H plus concentration ratio. Passing on, sources of acids in the body, we have volatile acids and non-volatile acids. So, volatile acids are mainly derived from the carbon dioxide formed during oxidation of the carbohydrates, proteins, and fats. These carbon dioxide forms as a carbonic acid which will be excreted through the lungs. Non-volatile acids are produced from the metabolism of protein containing sulfur containing amino acids like cysteine, methionine, lysine, adenine, histidine, and all. So these acids cannot be removed by lungs, but they are regulated by kidneys. What is the mechanism maintaining the acid base balance? So we have buffer system, renal mechanism, respiratory mechanism for this. Buffer system is actually for the immediate action. So here the H plus buffer resistant changes in pH by binding the free hydrogen ion. So major body buffers are bicarbonate buffer, phosphate buffer, and protein buffer system. So this bicarbonate buffer system is a important ECF. Its components are carbonic acid and HCO3, a base. Next is acts when strong acids or bases are added. Controlled by lungs and kidney. So carbon dioxide by lungs it is excreted and bicarbonate by the kidneys. Phosphate buffer system. So this consists of the sodium phosphate and acid and base. Coming to more powerful intracellular and in kidney tubular fluid. So important because its pH is 6.8 which is close to the body's pH. Coming to protein buffer system, most abundant buffer in the body present in blood and cells. Hemoglobin is the most effective protein buffer, especially in RBC. Deoxygenated hemoglobin acts as a strong buffer. What is the respiratory mechanism? This takes some minutes. That was an immediate thing. Okay, buffer system. Whereas respiratory mechanism takes some minutes to regulate. Like the lungs regulate the pH by altering the removal of carbon dioxide. So increase in carbon dioxide increases the number of hydrogen ion and decreases the pH, making it acidic. Hyperventilation removes carbon dioxide to decrease the hydrogen ion concentration. By increasing the pH, it makes it alkaline. So here this is the reaction. This reaction is reversed in lungs where CO2 diffuses out. Coming to renal mechanism, this is most powerful. It takes hours to days to return back. Kidneys maintain long-term acid-base balance by excreting hydrogen ion, reabsorbing and generating new bicarbonate. This mechanism corrects the chronic imbalances effectively. Next, water and electrolyte balance. So these three topics, antidiuretic hormone, aldosterone and thirst mechanism is also an important topic. Coming to this, water and electrolyte are the balance, refers to an regulation of body fluid volume and concentration of major electrolytes such as sodium, potassium, chloride, etc. So why it is essential to maintain the osmolarity, blood pressure, cellular function and also the homeostasis. That's why we study this under this topic. So this balance is regulated by three things: ADH, aldosterone and thirst mechanism. So let us see each one in brief. So, antidiuretic hormone, this is actually secreted by the posterior pituitary. What stimulates the secretion of this? Increased plasma osmolarity, decreased blood volume and pressure in the nephrons. Next is action. So, its action increases the water reabsorption in the distal tubule and collecting duct of the nephron by inserting the aquaporin channels and concentrates the urine, dilutes the plasma and restores the osmolarity. Coming to aldosterone, so this is a hormone secreted by the adrenal cortex, zona glomerulosa. So its stimuli are renin, angiotensin, aldosterone mechanism, and hyperkalemia is increased number of potassium in blood, and hyponatremia is increased sodium level in blood. So these causes the secretion of aldosterone. This will increase the sodium absorption and increase the potassium and hydrogen secretion, and it maintains the sodium, potassium and ECF volumes. Next, coming to the thirst mechanism, it is controlled by the hypothalamic thirst center in our brain. So, stimuli is increased plasma osmolarity, decreased blood volume or pressure, by the angiotensin 2, dryness of mouth. These are the stimuli of this centers. So, what it causes? Drinking water reduces osmolarity and restores the fluid balance. So, on summarizing these three things, we have ADH maintains the water balance, aldosterone regulates sodium and potassium balance, and what does thirst do? It takes control over water intake. So together they keep our internal environment stable. So basic components of food. So this is also a topic mentioned in the syllabus. So this generally we have studied in our smaller classes only. So why is this important? What our food contains? It contains the nutrients which are required for energy, growth, repair and regulation of our body. Then what it consists of? Carbs, proteins, fats, vitamins and minerals. So here we have macro and micronutrients. So macro are carbs, proteins, fats. Vitamins and minerals come under micronutrients. So carbs actually forms the 55 to 60% of our daily diet. Mainly, main energy source of our body is carbs. So it is about 4 kilocalorie per gram we take. It includes sugars and starches. So this is mainly going and assimilating in the form of glucose and stored as glycogen. So why it is essential for the brain and RBCs to prevent ketosis and spare the protein. Coming to proteins, it is formed of amino acids only, so it is composed of amino acids also. So this also provide 4 kilocal per gram. A need for growth, tissue repair, enzymes, hormones, antibodies, and it maintains the oncotic pressure and helps in acid base balance also. So per day, a person may require 0.8 to 1 gram or kg per day of proteins. Okay, so if a person is of 70 kg, he needs 70 gram of protein per day. This is a basic requirement of protein. This is a must. Passing on. Next is fats. So it is a concentrated energy source for 9 kilocalorie per gram. So they may include triglyceride, cholesterols and essential fatty acids. So they are required for the cell membrane insulation and absorption of fat soluble vitamins, that is A, D, E and K. So it provides 20 to 30% of our daily calories. Coming to the micronutrients like vitamins and minerals. So vitamins are organic compounds which act as coenzymes also. Okay. Next is fat soluble. A for vision, D for bone, E is an antioxidant, K is required for clotting. Water soluble things are B complex metabolism and C for collagen immunity. These are needed in small amount but essential for normal metabolism. So it is good that if you know the name of each vitamins and their functions, what they help for, and also the coenzymes, what they do us. Okay, this is actually important because when you go for your next year, you have to know all these things, which disease, what deficiency, all these things will be the base for treatments and diagnosis. Okay, coming to minerals, inorganic elements for structural and regulatory functions. So, here we mainly include the irons like calcium, potassium, sodium, magnesium, phosphorus. These are for bones, nerve conduction, muscle activity, and fluid balance. So, trace elements of iron, iodine, zinc, fluorine, they are found in our body parts like iron in HB, iodine in thyroid, and zinc as an enzyme, and fluoride in teeth. So, yes, by this we conclude the chapter. I have some more topics to be discussed in this chapter, which are actually not required for your exam purpose but for the knowledge sake, that I would be posting as a separate video on the upcoming days. So, keep following the channel and keep supporting. Have a great day. Thank you.